Continue to question 19. Now, Rosa conducted an experiment to compare the rates of evaporation of three liquids X, Y, and Z. Okay, he poured 10 cm cube of each liquid into three similar glasses. So over here, right from the top, we can note that your X, Y, Z is your change variable, and your measured variable is actually your rates of evaporation. Is what you want to find out now, right? Now, in order for this to happen, I'm just trying to unpack, thinking aloud for you. You cannot measure the rates of evaporation, but you need to measure something else, all right, in order to use it as a proxy for rate of evaporation. And in this case, the proxy, if you look at here, is the time taken for each liquid to evaporate completely. Okay? So, obviously, the longer time you take, it means that the slower the rate of evaporation. Now, give a reason of how each of the following help to make her experiment a fair test. So, fair test meaning one change variable. So, basically, remember, we need to actually talk about what happens when you don't. So using similar beakers, flip the question, what if you don't use a similar beaker? So you think about it, when you don't use a similar beaker, a few things can happen, right? And let's think about the factors that affect the rate of evaporation. You know, factors affecting rate of evaporation will be, let's do a bit of revision, okay? W, E, T. So W stands for wind, E stands for exposed area, T stands for temperature. So these are the three factors that you have learned. There is one more known as humidity, which we don't focus on, but basically these three are the ones that could affect rate of evaporation. So ask yourself, if I don't use the same beaker, which of these will it affect? And you will realize that the answer is your exposed surface area. Okay? So then you know that your answer, you need to talk about exposed surface area. So how would the answer look like? So it will ensure that all the beakers have the same amount of exposed surface area, and thus the rate of evaporation is only affected by the type of liquid, right? Next, placing all three beakers at the same place. So flip the question round. What would happen if you don't, right? So if you put him in different area, think about it in terms of environmental conditions. And then you look at this, what affects rate of evaporation? Okay, you can have wind, you can have temperature. So this too is actually factors associated with your location or the type of place that you put them in. So if you don't put them in the same place, these two might differ. So by putting them in the same place, it will ensure that the surrounding temperature and the strength of wind is the same, right? So can you see now how does flipping the question actually help me with my answer? So it will ensure that all the beakers have the same surrounding temperature and strength of wind. Hence, the rate of evaporation is affected only by the types of liquid. Okay? Now, moving on to the next question. Yan Ling set up an experiment as shown below to compare a property of three strips PQR, which are made up of different materials. So right off the top, we already know that the change variable is the type of materials. For each strip, she added 50 cm³ of water to the container and measured distance d. Her results are shown below. So this would be her CV and this would be her MV. Okay? Now, state two variables of the strips that are fixed. Fixed, that means, okay, the same or constant. Okay, such the experiment is a fair test. So you think about it. Sometimes there are many things you can talk about because basically everything else needs to be the same except the change variable. But sometimes some answers have a higher quality than the others or are more relevant. Okay, so for example, if you take a look at it, what they want to find out is basically what is the distance over here, which means how much your strip is bending, correct? So you would ask yourself, what could affect the bending distance of the strip? So in the first place, for example, okay, length of the strip, right? We know that the longer the strip is, and then you put an object, a weight in the center, the more it's going to bend. The thickness of the strip, correct? The thickness of the strip. The thicker the strip, most probably is not going to bend so easily. So these are qualities or variables that could affect the bending distance of the strip. But of course, you know, you would argue other things. For example, you may say things like, oh, the type of wood to make the wooden block, the height of the wooden block. I would agree. Yes, is it a correct answer? Absolutely. But is it the best answer, the answer that the examiner was really hoping for, that affects the experiment directly? No, it doesn't. Okay? But to be a fair test, everything needs to be the same except the change variable. But there are certain variables that are more important. So, and in this case, they did explicitly talk about the strips. So you have to mention about the strips. Okay? I'm just pointing it out that some students usually just put their answers, all types of answers. Okay? So how would my answer be? So I can say length, I can say surface area, I can say width size, I can say thickness of the strip. Okay, so all this should be taken into consideration and they need to be constant. Right, moving on. Next, Haziq used the setup to find out how the two type of bulbs affect the temperature of its surrounding. So 2 type already tell me that this is the change variable, okay, and my measured variable is probably going to be temperature of surrounding. Okay, now bulb R was lit up. He covered the box and recorded the temperature of the air, okay, over a period of 10 minutes. He then removed bulb R, left the box uncovered for 15 minutes before repeating the experiment with bulb S and then T. The results are shown in the table. Okay? So give a reason why Haziq left the box uncovered for 15 minutes before repeating the experiment with the next bulb. This is actually a fair test question. Okay? A fairly common fair test question. So the whole idea now is, remember I told you, you should flip the question. What happens if you don't wait for the 15 minutes? So if you realize that if you don't wait for the 15 minutes, okay, you would see that after 30 minutes, okay, the temperature of R is at 30 degrees, correct? And straight away you put in S. What will be the starting temperature? Instead of 25, it will be 30. So then you realize that the starting temperature is not the same. So remember this. For fair test, all variables must be the same. Okay? So by flipping that, you realize that, and your answer become very easy. So it is to ensure that the beginning or the starting temperature of air in the box is the same at 25 degrees Celsius when the experiment is repeated for the next bulb. This will ensure that the experiment is a fair test as there is only one change variable, okay, where the only change variable is the type of bulb. Okay. Right. Next, we're moving on to something new. It's called experimental and control setup. Alright? So what is a control experiment? So a control experiment is an experiment where no variables are tested. Okay? It is an experiment, or in contrast, an experimental setup is where a change variable is tested. Okay, so what is the purpose of setup A? So this is a common question whereby they always ask you, oh, what is the purpose of setup A? Actually, the answer it is a controller, right? So how do you then answer it? You need to memorize this whole framework. So it acts as a control setup to compare and confirm that the measured variable is only due to the change variable and not whatever else is there, okay, in the question. Okay, and not to the surrounding air temperature, not to the light intensity. It depends on the question. So what do I mean by that? Let's move on. Now, let's look at this. Martha wants to investigate if the number of leaves on the plant affects the amount of water taken in by the plant. So this is actually your change variable, right? Okay, everything else needs to remain the same. So Martha has 3 sets of similar plants and 3 500 ml glass beakers. She may use some or all of the items. She filled 3 glasses with 400 ml water. What items should she put inside the beaker for each set up? Okay, so basically the change variable in this case, alright, you will need to actually be number of leaves. Okay, so remember, a control setup is where it's a setup with no variables to be tested. So in this case, the one that you want to test is actually your number of leaves. So very simple. So she needs to have, to set up a control setup, she needs to have a setup with no leaves. You remove all the leaves. That is your control. Okay? So what is beaker 1 and what is beaker 2? So for your experimental setup would be a plant with all the leaves intact. That means you don't remove any leaf. The second experiment will be a plant with fewer leaves than beaker 1 intact. So let's just say beaker 1 has 10 leaves. Okay, your beaker 2 must have lesser leaves. And then your control setup is totally no leaves. That is your control setup. You remove away the change variable. The variable to be tested is gone out of the equation. So far okay? So let's take a look. Junrong wanted to find out whether germinating seed produce heat. He soaked some seeds in the water before placing them into a glass container as shown. Suggest a control setup to show that the rise in temperature was caused by germinating seeds. So in this case, okay, what was the variable, the factor that is being tested here? What is the change variable? Is there even a change variable? Actually there is, right? Germinating seeds. Think about it. It's either you have or you don't have, right? If you think about it, Junrong wanted to find out whether if you change the wordings the presence of germinating seeds produces heat. If I change it to the presence of, so then you realize, the presence means have and don't have. That's the difference, right? One scenario have, one scenario don't have. Correct? So actually this is so-called like your change variable or your variable that you are testing out. This is the variable actually. So a control setup means that you want to actually remove away this variable. So in your control setup, there will be no seeds. You do not have the soaked seeds. Okay? So what you would do is you would prepare a similar setup as shown in the diagram. But without the soaked seed. That would be your control setup. So for example, let me give you another example to just to cement this idea. Say you want to actually find out if drinking more milk makes you taller, you will need a total of triplets. So they need to be genetically identical. They will be subjected everything to the same. Sleeping time, studying time, what food they eat. The only difference is that the control will not drink any milk because the change variable is the milk. 0 milk for triplet number 1. Number 2 will drink some milk. Okay, and number 3, they will drink more milk than number 2. Alright? Okay, now, so basically, Jill conducted an investigation. She took 2 similar pots, A and B. She put a plastic bag around the pot of A, as shown below. She tied a string tightly around the plastic bag and the stem. She placed them at the same place and watered only the leaves of both plants. What is the purpose of plant B? Okay? Alright, so plant B basically is a control. Right? Okay? So it acts as a control setup to compare and confirm that growth of plant is only due to the roots absorbing water and not the leaves. Okay. Next. This is an actual PSLE question. So, sorry, I forgot to tell you all that, sometimes the question will tell you from which year the PSLE question came up from. Unripe bananas are green and contain chlorophyll. Ripe bananas are yellow and give out gas X. Priya wants to investigate whether gas X will help unripe bananas to ripen more quickly. So actually, this is the one that we want to investigate, right? So this is also like our change variable. Okay? So she has only the items below. She may use some or all of them. Okay? Priya used 2 identical clear containers for investigation. What items should she put inside the container for each setup? So what it means is that, okay, she basically has this amount, okay, 1 experimental setup and 1 control setup. So what should she use? So think about it, right? You want to actually compare what is going to happen. So experimental setup, you want to see whether gas X will help or not. So in your first experiment, what do you want to put in? for your experimental setup. You would have used up one unripe banana, okay, and one ripe banana. Okay. Why do you think you need to actually have the ripe banana with the unripe banana? Because you want to find out whether is it ripe, does it ripe faster, right? So obviously you need to have an unripe banana. So your ripe banana there, in this case, right, is a trick question. It's actually to provide the gas X. So without your ripe banana, if you don't have it there, how are you going to get gas X, right? So cannot. So this is actually a trick question. Many students just gone case because they didn't realize that they need to put the ripe banana, because the ripe banana is actually gas X. It's really literally a trick question, dumbfounded. Then your control setup, remember, you want to remove away the change variable. So in this case, your change variable is gas X. So all you need to do is to put one unripe banana to stop. That's it. Because without your ripe banana, you would not actually have your gas X. And then you can compare whether or not your unripe banana and your unripe banana with your gas X, what is the timing when it comes to ripening. So once again, let me explain. So a lot of students didn't realize that they needed one ripe banana in the experimental control setup. And the function of the ripe banana was not really because it was a ripe banana, or wasn't to compare anything, because this is already ripe already, is to give up gas X. Yeah. PSLE question, this is the kind of level. Next. Hypothesis. Okay, now it is a prediction of an outcome based on scientific reasoning. Okay, so how do you actually state a hypothesis? So you basically would say, if don't know whatever change variable increases or decreases, then the measured variable should increase, decrease, or remain the same. Or if it's something that is present or absent, like for example in the previous one. So you will say, if gas X is present, or if ripe banana is present, then the ripening time of the banana should increase, decrease, or remain the same. Now, this increase, decrease, remain the same, you may think it's very silly, but actually there is no wrong answer as long as your structure, the way, the cause and effect that you answer it is correct. Because an hypothesis is something that scientists come up with before they carry out an experiment, which means it can be correct or it can be wrong. So this is based on their reasoning. So for example, let's just say, because I know from the word equation of photosynthesis, light is needed. Therefore, if light intensity increases, then number of bubbles produced for a water plant should increase based on my knowledge. So that is correct. Whether is it right or wrong, it doesn't matter. I can also say, if light intensity increases, then my bubbles for water plant would remain the same after a certain period of time. Let's say 24 hours. Whether is it true or not, I don't know. But maybe because I feel that my hypothesis is all the chloroplasts will have been fully utilized. So even though I keep increasing my light intensity, it will remain the same. Okay? So let's take a look. Mr. Cole placed 5 identical fruits, okay, A, B, C, D, and E, in rooms of different temperatures and measured the time taken for each fruit to ripe. The results of his experiments are shown. State the hypothesis. So take a look at it, okay? You have different temperatures. This is something that you change. This is your change variable, right? So I would say as the temperature of the surrounding increases, the time taken for the fruit to split should decrease. Something like that. Or I can say should increase. I am not going to look at this to write my hypothesis because it shouldn't be the case. This is results. Your hypothesis comes before your results. And then you carry out your experiment to see whether or not your hypothesis is correct or wrong. Okay? But I can say that if the change variable, which is temperature of surrounding, increases, the MV, which is measured variable, time taken for the fruit to split should increase. So this part should increase. Alright, it's like something like maybe say you draw inference, because in during science lesson your teacher tells you already for a fruit to split it needs to dry by explosive mechanism, right? It needs to dry. So then you think, okay, with increasing temperature, evaporation happens at a faster rate, so my seed should dry faster. Okay, so it should take a shorter time to split. Okay, so that is based on your inference. But whether or not it may not be true, maybe there are some weird species that doesn't adhere to that. Question 27. Therese set up the following experiments. She placed a light sensor at different distances, okay, X from the torch as shown below. Okay, state the hypothesis. So what's the change variable here? Different distances. Okay, so I would say as, okay, or rather if the change variable distance of light sensor from the torch increases, the amount of light detected by the light sensor should increase or should decrease. Up to you. Okay? So really, in reality, this comes before your results. But of course, if the results is there, then you follow suit. Okay? As you can see, this one, right? As distance x increases, your amount of light decreases. Okay. Next, David studied the effect of surrounding temperature on the life cycle of organism G. His findings are shown below. State the hypothesis. So once again, if change variable, the temperature of the surrounding increases, the MV number of days for one complete cycle, life cycle of organism G should increase. Okay? So really, hypothesis is really that framework that you need to follow to write. That's about it. Okay? Your cause and effect needs to be correct arrangement. As long as that is in correct arrangement, that you cannot really be penalized. Alright? So you can say if the temperature of the surrounding increases, the number of days should decrease or something like that. Okay? But of course, you know, when the results are shown, you follow the results and you use the result to deduce the hypothesis. You understand? You use it. Okay? So because from here, you see that it's actually as temperature increases, life cycle days actually decreases. So you use this as evidence to support your hypothesis. Okay? If it's given. In fact, if it's given, you must use this. Okay? That's what I'm saying. But in real life, for hypothesis is based on research and findings. So you take these as your research and findings to deduce this. Understand? Must, okay? You don't later on say, Mr. Chong say, anyhow write also can. Whether this one increase, decrease, then I know. If they give you the results, you have to follow the results that is given. Okay? Got it? So it's a little bit like a relationship question, right? You would say, so the difference is this. The difference is that the question that will not say, what can you conclude? Or what is the relationship of temperature with the number of days of an organism? Then your answer will be, as the surrounding of the temperature increases, the number of days for one complete cycle of the organism G decreases. Okay, it's very similar, but it's not. Okay, next. Generating possibilities. Now, so this is the scale of exploring all alternatives. Now, whatever questions are here, PSLE question. So, I tell you, you need to know. Alternatives, possibilities, and choices beyond obvious or preferred one. So this is where you need to think out the box. Okay? Because we've been exposed to so many experiments, many of times we think that the answer is like that. So this is really to test whether or not you can think beyond the common, the typical answers. So for example, Mary conducted an experiment to find out if different wind speeds, okay, this one over here is a change variable, affect the distance moved by C, X as shown. So this is actually your measured variable. Now, Mary repeated the experiment 6 times with all variables kept the same except for one. Suggest 2 possible ways of varying this variable. So, the variable that you want to change will be different wind speed. So now they're asking you to think out the box there. Hey, how can you change the wind speed? How?
You use your mouth.
Okay, so basically, okay, you can change the speed of the fan, or you can add more number of fans. Okay, so you can do that. That's fine. So you have to think out the box. Okay, what else? Another one. Cell X is an organism that has the features of both an animal and a leaf cell. When cell X was viewed under the microscope, it was observed that part P contain a green pigment which could be found in all green parts of a plant. The eye spot of the cell X can help guide the cell to move towards a light source. Suggest how this ability enabled cell X to survive. So basically they're asking you to suggest more than one way. So, but most obviously, you always look at the question stand for hint. So the hint over here is green pigment. So you ask yourself, green pigment, where did I hear this word? Under the topic of photosynthesis. But of course, if you never listen to your teacher, then I cannot help you. So basically, a green pigment signifies probably chlorophyll. And if you can have chlorophyll, you basically can photosynthesize. So in order to photosynthesize, you will need light. So that is where this light actually comes in. So when it's able to help your animal cell to move towards the light source, it will enable it to increase in photosynthesis. Now be very careful. Students, this is extremely pertinent. I have repeated myself many times, but yet I still see the same problem. Many of you would say things like, oh, the light spot will help the plant to find light, therefore enabling it to photosynthesize. But in actual fact, that when you say words like this, it means that without the eye spot, the plant cannot photosynthesize even with the most minimal amount of light. That's not true, right? Okay? As long as there's some light, you can photosynthesize. So what the light spot does is that it helps it to find more light, hence enabling more photosynthesis. So it's really about getting more light for more photosynthesis. Okay? So the eye spot help cell X to find more light for more photosynthesis, to produce more food for growth. Okay. Can you see how it's very nicely related? More light, more photosynthesis, more food growth. Now, why not more growth? Because growth in itself is increasing in size already. You realize? Okay? Alright. Last but not least, we have arrived at a very, very important. So we've covered fair test, we've covered reliability. Now we're finally at accuracy. So accuracy is about the measured variable, okay, obtained precisely. Now, so let's take a look at this. Lucas conducted an experiment in two similar rooms as shown. He used a data logger to measure the temperature of the two rooms every 30 seconds for 2 hours. Why did Lucas use a data logger to measure? Okay, the data logger, use a data logger to measure temperature instead of a laboratory thermometer. Okay, to visually record the temperature every 30 seconds, right? So basically, the data logger is more accurate. It's by computer. So basically, they will be very on the dot when it comes to measuring. That's one. You reduce human error. The second thing is that the decimal places. So maybe say a double laboratory thermometer can only measure up to 0.1 accuracy. So for example, 31.1. Whereas a data logger is 2 decimal places, 31.15. Okay, it is more accurate in that sense. So using a data logger will have a more accurate measurement of the temperature. Okay? So what else? It is equivalent to like if I ask you to measure a running track, the length of a running track. I give you a measuring tape and I give you a meter rule. The measuring tape is more accurate, okay, because it's continuous as compared to the meter rule. Okay, next. 32. John placed the same amount of seawater which contain living organisms into two identical glass containers, A and B. He placed container A into an opaque box while B was placed under a brightly lit room. Every 6 hours, John opens the lid to measure the amount of carbon dioxide in each container. His results are shown. Okay, suggest one possible reason that could have resulted in John obtaining inaccurate results. Okay, so remember, when it comes to accuracy, it's got to do with how precise this person in obtaining the measured variable. So the measured variable was the amount of carbon dioxide. Now you think about it, okay, as one of you rightly pointed out very loudly, when you try to measure it and he opens the lid, right, you realize that every time he opens, the gases exchange, correct? Some of the air inside the lid could go out and some of the air from outside surrounding it go into the beaker. So it's not accurate, alright? And he does this continuously, for every 6 hours. So you can imagine, okay, it is not just a one-time error, it could be as many times as he opens the lid. So what would be more accurate, okay, would actually be to put in a data logger inside there permanently, okay, and seal up any opening. Alright, if they ask you for a solution. So what would the answer be? So when John opens the lid to measure the volume of carbon dioxide, some of the gases may have escaped from or entered the container. So in this case, we are actually concerned about carbon dioxide, right? So you could technically replace this answer with carbon dioxide since we're only concerned with carbon dioxide. So I would just like to stress that, the three things that we are really, there are actually 5 things that we want to look for in an experiment: change variable, measured variable, okay, and then control. On top of that, we also want to look at what is reliability, fair test, and accuracy. Make sure you know that reliability talks about how many times can you obtain similar or the same results. The more number of times, the more reliable. Fair test talks about only having one change variable and the rest being the same. And lastly, accuracy talks about how precise can you get your measured variable. With that,